we are going to present the app we made. We call it as SMAP, Standard Microarray Analysis Pipeline. And it's an R-Shiny educational app for transcriptomics analysis. We basically had 5 groups: Group A, Group B1, B2, C, and D. Group A worked on the backend of the app, developing the transcriptomics pipeline. B1 worked on the app functional components. Group C basically worked on GitHub management and creating the documentation for the whole app. And Group D worked on developing a project site as well as logging the intern experiences as well.
So we already loaded data in, but basically we're using raw data from a lung cancer study that looked at normal and cancerous tissues patients. And in addition, we have a metadata file that we uploaded that basically has the sample names along with the condition status of whether the tissue has cancer or is a normal tissue. Here, there are like 2 primary things we can do. Before conducting statistical analysis, if we had a data set where there were a lot of genes that are expressed at very low levels and we're concerned about the quality of that expression data, we can filter out a certain percent of it, like 1 or 2%, and hit the filter out genes button. That wasn't really an issue for this data set, but we have that option available to us. And so then after deciding what we want the cutoff p-value to be, we can hit the find differential genes button, and then you're given a table with the fold change values and the adjusted p-values for all of the genes. And then to more visually understand how many differentially expressed genes we have, SMAP also has a volcano plot. And here we can change the log fold change cutoff value as well as the p-value, and we can adjust how many differential genes we have.
So SMAP offers 3 different types of visualizations for the functional analysis step. We can start with the KEGG pathway analysis. And as you can see here, if you scroll down, users can choose to analyze using 2 different types of visualizations, the dot plot and the bar plot. And further, they can adjust the number of pathways depending on the number of significant pathways that they would like to isolate for further analysis. And last but not least, the user can also do the GSEA analysis, which if you click that button, the plot shows up below as shown. And here we have the FAQ tab, and since this app is for educational purposes, we have a very comprehensive collection of FAQs to help students understand places of the pipeline where it may be unclear to them.
So not only did our team build a full functioning app, but we were also able to create a documentation site and a tutorial page. The documentation includes things like code description, significance of the output, and important functions for all the functions our app has, like data importation and normalization.
We use diversity as our strength by engaging with our team members and our mentors and learning how to code, as well as use online resources such as documentation to develop our code. Some skills that we developed in this process was building an app layout, as the STEMaway platform only provided us with the transcriptomics pipeline part. So we worked with the UX team in a cross-pathway collaboration at STEMaway in order to develop this layout. In addition, we learned to use other apps and resources such as Adobe Illustrator and GitHub.